Okay, I guess we're live. Maybe I should button this up. Try that. Okay, let's see. So everybody supposedly has put in their paragraph. So the papers should be somewhat chaotic now, but they should have something there. And then there should be references at the bottom. So here's an example, and you can see who your co-authors are, right? And then you could read down and try to see what's happened. You could probably get rid of this 1, 2, 3, 4, 5. Just make them in alphabetical order. And Paul could be abbreviated P. Period. Wow, it seems a little. Does this work? No, that doesn't work. Okay, whatever. So you can kind of fix up some of the easy stuff to fix up. That's fine. But the main thing to do first, before you start changing everything, is to go to File, see Revision History, and then kind of click through and see who's done what. So see who's added what, and well, that one wasn't much of an ad. Let's see, how about this one? They should be in different colors depending on who added what when. And you can then go through and figure out who did things. And then maybe take a little piece of paper, like real paper, and write down, oh, I think this person did a good job here and there, that, the other thing, right? And then go to Moodle, and in this week's Moodle block, there's Peer Evaluation 1A, so Paper 1, Evaluation A. And you click on that, and then choose your name and put in the last 3 digits of your ID and choose your topic. And this will take you off to where you can fill out a little rubric. So for each person, you're basically giving them a grade, and the grade should be based on how well they did the assignment, which doesn't mean, the final paper. It's just, did they find a reference that was really good? Did they write about that reference in a useful way, that gives you something to work with? Is it in proper college-level English? That type of thing. And I would say, don't just, fire off A's, but also don't be super harsh. You want to be treated. You want to treat other people as you would like to be treated, right? And it's a tiny amount of your grade, but this builds up over, the semester. There's all these different peer evaluations. Okay, so then after you've done that and you've submitted your peer evaluations, then go back to the paper, go back to the Google Docs here, and start adding comments. Because the next peer evaluation is going to be on how helpful you were in fixing up the paper. So start adding comments. You might also add sentences here and there, like maybe you read something that now pertains to a paragraph that you didn't really originally enter. And you can, insert a little sentence there talking about, how your reference also is relevant. And just start getting the paper into order and start sending comments to your co-authors, telling them, what you think and why you think, how you think it should be improved, and try to be a good teacher. Like, the best way that you can learn is to teach. And so try to teach your co-authors what your thinking is at your stage. And this is not really that different than the way I write papers. Like, I write papers with people who are all over the world, and I almost never see them. And we just, throw stuff into a document, it churns around for a couple of years, and then it's pretty much ready. So, try to do the same type of thing, and hopefully this will all just be great. That's what I'm thinking. So try to take care of your peer evaluation the next few days, get your comments in, and then, like after next Sunday at noon, then the next peer evaluation will open up for how you've commented on the paper. And then you'll still have some time after that for the final fix-ups. All right. Let me out of here. All right, so any questions or comments? Yeah, go ahead.
Quick question on paper. I know we're all supposed to have like our own article. Is it okay if like all of our articles almost are all from the same author, just different articles?
Yeah, but they're different articles? Yeah, sure. Some people devote their whole lives to a subject, and they've written lots of things, and they've kind of built up. Yeah, so it's fine if, and I expect that will happen, maybe not all, but there will be multiple articles from an author or set of authors very, very frequently. Yeah. Mm-hmm. Yeah. I think so. After the quiz is closed, after everybody's had an opportunity to take it, then you can click on your score, and you go back to the quiz, and you click on your score, and it should give you the quiz with the correct answers. Yes, I think so.
If that doesn't work, you click on attempt 1. You just click on the 1, and it will take you to the quiz.
Oh, okay. Yeah. Yeah. Can I use the book? Can you cite the book in your paper? Yes. Yes, you can cite the book in your paper. Or any other book. And what I don't want is like Wikipedia and sort of stuff that's very derived that you could never trace back to the original source. Yeah. Okay. Let's see. We had a few questions, but I don't really know what to do with them. Maybe we'll just keep on going. Okay, so what's going to happen in the next few lectures is we're going to march through chapters 7, 8, and 9, and then I think that's all the chapters for the big test. And this is like it's maybe not the most cutting-edge stuff. It's stuff that has been in textbooks for a while. And it's kind of the background that I think we need everybody to get up to before we can really delve into interesting sort of side topics. So you might think of it as the core of evolutionary biology. And what's going to happen today is I'm going to start out with some mathematical models, very simple mathematical models. You might even think of them as simplified mathematical models, where you try to distill down the essence of the process. And then we'll build on those as they progress. It'll also develop a lot of our vocabulary and just sort of the way that we speak about things. And there's a lot of pink slides, but I don't know if they're terribly difficult pink slides. It's just like every section in the book is adding some information that everybody needs to know. Okay, so this first chapter is on the genetics of populations looking at a single locus. So if we just focus down to one locus at a time, then how can we understand lack of evolution and then the various forces that might affect evolution? And it all sort of hinges on this model called the Hardy-Weinberg model, which you've probably heard of before. And this Hardy-Weinberg model, it has several what you might think of as assumptions, and then it has some outcomes. So like one of the outcomes is that dominance by itself doesn't cause evolution. And one of the assumptions is no selection. Then after we've built up that, after you understand that, then we'll start adding things. So we'll add selection, and then we'll add mutation, and then we'll add selection and mutation, and so on. Okay, so maybe the first thing to note is this difference between what you might think of as a discrete trait and something that's a continuous trait. And a lot of our continuous traits are polygenic. They're caused by lots and lots of genes, and actually also lots of things about the environment. So skin color, of course, is a polygenic trait, or height, or almost any dimension of a vertebrate animal would be a continuous trait. And then a discrete trait is whether these plants rotate clockwise or counterclockwise. Now, we think that both the continuous traits and the discrete traits, to the extent that they're underlain by genetic causes, those genetic causes are discrete. There just might be a lot of little discrete genetic causes, and they kind of all add up to having an effect. So, yeah, go ahead. Yeah, so in the genome, there's a whole bunch of loci that affect skin color. At each of these loci, there's variation in the population. And that variation is ultimately discrete because it's based on allelic differences, and the alleles are one way or another. It's just that all of these little discrete things add up in kind of a voice-vote way to determining what the height of a person will be. Yeah.
Or like the knot in your head, like your hair knot. So there's like that spiral knot.
Yeah, so the spiral knot might be a lot like these plants, right? And the character might be discrete, but that doesn't mean that it's necessarily based on a single locus, right? Because it could be that there's a whole bunch of loci that affect, the spiral on your head, and it's just there's like a threshold point where if enough of the loci are screaming left, then it goes left, and enough of the loci are screaming right, it goes right. Yeah, go ahead, Mike.
The continuous would be like add loci, focus loci, or no?
Say it again?
The continuous trait, would that be found like at a focus loci?
Locus? I think for continuous traits, our usual interpretation is that there's multiple loci involved. Although, of course, there also is likely to be multiple alleles at any one locus involved. So, for instance, let's say we had some locus that is basically a step in a biochemical process that makes an enzyme. Then at that locus, you could have dozens of alleles, and those alleles could vary in how good makes the enzyme work at cold temperatures versus hot temperatures, right? And so that also contributes to the continuity of the variation. But at our core, we believe that the inheritance is not just blending inheritance. It's not just if you pour black paint and white paint together, you get gray paint. That at its core, there's all these little teeny discrete effects, or Mendelian effects, and that because of there being many of them, it appears to be continuous. But it's inherited in a Mendelian fashion. And by inherited in Mendelian fashion, that implies that as the generations march on, you're not changing the alleles by virtue of mixing things up. The alleles are staying the same. All that's changing is the composition of the alleles in the individuals. Yeah. So that was what Darwin didn't understand, right? He didn't understand the nature of variation. Deep down is a discrete thing, the nature of genetic variation. Anyone else? Okay, so having said that, then I guess we'll sort of plunge in. The other thing that you have to think about here is that we're really thinking at the population level. And this is something that Darwin was very good at. So we're not focusing in on individuals here. We're trying to understand how populations change or don't change. So individual thinking would be like what gametes and offspring are produced in what frequencies from a given pair of parents. And you might do something like that Punnett square up there. So remember, Mendel would take individuals and mate them with other individuals and then get F1s. Then we take these F1s and mate them against one another and get an array of F2s. that's really all focusing on those two original parent individuals. And when you're getting an array of F2s, then assuming nothing is going on, just the standard Mendelian stuff, then you'd expect a ratio of 1 to 2 to 1, you know, for something that's inherited where you can see the genotypes pretty well. Now, with population thinking, we're not taking particular individuals. We're taking the population as a whole and asking, how does it change from one state or one generation to the next, or not change? And so it doesn't start out with this nice little equal balance where you have two inbred lines. Instead, it's just a population at whatever state it's at, and then it moves forward in time. So we're asking, how do characteristics of the population change over time as a result of these evolutionary processes? Now let's break this population thinking down to not just the diploids that we see running around. Like, you know, if you look at a population of squirrels, then those are all diploids. But we can also think about the allele frequencies in the gamete pool. So here we might have parent squirrels, and then some of those parent squirrels are A1, A1, and some of them are A1, A2, and then some of them are A2, A2. So those would be the genotypes. And if you only had two alleles, then you'd only have 3 possible genotypes. If you had 3 alleles, you'd have more possible genotypes, diploid genotypes. And you could calculate the frequency of A1, A1 in the population, the frequency of A1, A2 in the population, the frequency of A2, A2 in the population. Now you can, so those are the genotype frequencies, the diploid genotype frequencies. Now you can also think about the allele frequencies in the gene pool. So for each individual that's A1, A1, then the only kind of gametes that it can make are A1 gametes, right? The only type of gametes that an A2, A2 individual can make are A2 gametes. But a heterozygote that's A1, A2, half of her gametes are A1 and half of her gametes are A2, assuming no funny business. And similarly, if you had more alleles, like if you had A1, A3 individuals, then half of that of those individuals, half of their gametes would be A1 and half of them then would be A3. So allele frequencies we usually denote as P and Q. Like the allele frequency of A1 in the population is P, and the allele frequency of A2 is Q. And that's one thing to think about. And that's different from thinking about the diploid genotype frequencies. And you can get from one to the other pretty easily. So if you started with the diploid genotype frequencies, you know, say there were 60% of individuals were A1, A1, and 60, and then 20% of them were A2, A2, and 20% of them were A1, A1, then you could figure out what P is, right? Because 60% of those, 100% of the 60% are going to contribute A1 alleles, and then there's 20% of them are like this, and half of those are going to be A1 alleles, you know, so you end up with 70-30 in the population, in the gene pool. And then we could imagine that from this gene pool, this big pool of sperm and egg, come together gametes at random, and then they would make new diploid individuals. So here is this offspring generation that are diploid, and again, they each have 2 gene copies. And if this was all at random, drawn at random, then they would follow certain frequencies. And those are called the Hardy-Weinberg frequencies, which we'll get to in a second. Now, this figure was a little bit helpful, so I thought I'd throw it in, even though it's in one of the little math boxes, which I usually ignore. But in this figure, we have the frequency of A1 alleles in the population. It could go from 0 to 1, right? You could have no A1 alleles in the population, or you could have all of the alleles in the population be of A1. And then there's this other frequency, and that's what we'll call Q, which is the frequency of A2 alleles in the population. And similarly, it would go from 0 over here to 1 over there, right? Like if the frequency of A1 alleles is 1, then the frequency of A2 alleles is 0. And if the frequency of A1 alleles is 0.2, then the frequency of A2 alleles is 0.8 in this very simple model, because we're assuming in this very simple model that we just have 2 alleles. So P plus Q is equal to 1 if there are 2 alleles. Now, you wouldn't necessarily have to have 2 alleles. I'm just playing around with 2 alleles because the math is easy. But in a population, you could have, a third allele and a fourth allele and so on. And if you had that, then the sum of the frequencies of each of the alleles would add up to 1. And at this Hardy-Weinberg equilibrium business, then the relationship between P, or Q, it doesn't matter, and heterozygosity follows this line. So that's heterozygosity on the vertical axis. That's the frequency of heterozygotes in the population, A1, A2. That is, individuals that have different alleles in their same body. And it's bound on this line here. It can't be anywhere if it's at Hardy-Weinberg equilibrium. Okay, so what is this Hardy-Weinberg thing all about? So Hardy-Weinberg is like a null model. It's what would be happening if there were no selection, no assortative or disassortative mating, that is, individuals mate with one another at random in the population, no mutation, no migration, that is, no immigration or immigration, and no gene flow, and the population size is effectively infinite. Those 5 things are considered the only forces of microevolution. So microevolution is change in allele frequency at a locus within a population. And the only things that would cause change in allele frequency, that is microevolution, are those things, like selection, some kind of assortative mating, mutation, migration, or genetic drift in a finite population. And if we went through this little process of going from parents to gene pool to offspring, and there was random mating, then what would happen is that we would find that this equilibrium would be reached in one generation, just in one generation, if there was no overlapping generations. And if generations overlapped, then it might take a couple generations, but it wouldn't take a long time. And the frequencies would be p squared, 2pq, and q squared. So let me think out loud how that comes about. So we have this gene pool of sperm and egg. You reach in and you pull out a sperm, and the probability that it's going to be A1 is P, right? Because the proportion of A1s that are in the sperm pool is P. The proportion of sperm that are A2 in the gene pool is Q. So the probability that you're going to get an A1 if you just reach in and pull out a sperm at random is going to be P. Similarly, in the pool of gametes that are eggs, the proportion of them that are A1 is P, and the proportion of them that are A2 is Q. So if you reach in at random and pull out an egg, it's going to, the probability that you're going to get an A1 is P. So the probability that both of them will be A1 is P times P, or in other words, P squared.
Shouldn't the last one be A2 equals P squared?
Oh, yeah. I don't know how to change it. Is that... yeah.
How is this model useful if it's based on assumptions that don't actually exist in real life?
So no model is complete. No model that like completely captures everything that's going on in real life. A null model is one that you set up where you assume something that you don't actually think is happening. And then you can compare real data to it and try to figure out the way in which it deviates from the null model. And so then you would be able to infer some of the things that might be going on. So say we took a population of mice, and you went out and you sampled their genotype frequencies. Then you might find out, for instance, that there's way more heterozygotes than there ought to be according to Hardy-Weinberg. And then you want to know, well, why would that be? Why is that the case? So there's several possibilities. One is that it could be selection is favoring heterozygotes. So you might find more heterozygotes in the population just because the homozygotes are dying off. Another possibility is that there's disassortative mating, where there's more heterozygotes in the population because individuals avoid mating with other individuals like themselves, and so on. Yeah, go ahead.
Usually at the near end of the population, the allele frequencies would be equal. Or
What? Say it again?
Frequencies at the end of the population in time would be equal. Or
No, not necessarily. So if it so let's say we started out with a population where you had two alleles, and the frequency of the A1 allele was 40%, and the frequency of the A2 allele is 60%. So it adds up to 1, right? Yeah. If you didn't have any selection, and you didn't have any mutation, and you didn't have any of those things, then it turns out that no matter how long you go out, you'll still have that same allele frequency. It'll still be 40, 60, and the diploids in the population, the diploids would be of certain frequencies. So the frequency of A1 would be 0.6 times 0.6, which is 0.36, right? The diploid frequency for A2 would be 0.4 times 0.4. What? Oh, 0.16. You can think faster than I can. It's because of the microphone. It makes me think slowly. 0.16. 0.16. 16 out of 100 individuals. Yeah. And then the frequency of heterozygotes in the population would be 0.6 times 0.4, which is 0.24, times 2, which is 0.48, right? And the reason why it's times 2 is because it could be that you got your A1 allele from the sperm and your A2 allele from the egg. That's one of the PQs. And the other PQ is that you could have gotten your A2 allele from the sperm and your A1 allele from the egg, and that would be QP. And so PQ plus QP is 2PQ. So P is the frequency of the A1 allele in the population, regardless of whether they're in heterozygotes or homozygotes. And Q is the relative frequency of A2 alleles in the population, regardless whether they're in homozygotes or heterozygotes. And of course, if you had more than 2 alleles, then you'd have to add an R or you know, something like that, right? Or you could go, you know, P sub 1, P sub 2, P sub 3. Yeah.
So if you added the third, like allele,
Third allele.
It would be like A1, A2, A3. It would be 2 PQ R if you were to do R on top of that.
Yeah.
Okay.
Yeah, basically. Yeah, so you'd have to figure out each combination. So if you had 3 alleles and one of them, the third one, it occurred at frequency R, then the frequency at which you would find individuals that were A1, A3 would be P times R times 2.
But never like P times Q times R.
No, because everybody's diploid. You're imagining some space aliens that are triploid. Right.
What if it was triploid? What if it wasn't?
No, you mess up my brain. Okay, and then the other, in the back. The next thing is dominance doesn't cause evolution. So all of this is true whether A1 is dominant or A1 is recessive or A1 is codominant. It doesn't matter. It's not the case that the dominant allele by itself would evolve to become the most frequent one. Dominance and high frequency don't cause one another exactly. So it's not the case that as time goes on, we're getting more and more dominant alleles. You know, not because of just having sex. Sex by itself, meiosis and fertilization by itself doesn't change allele frequencies, and it doesn't favor dominant alleles over recessive alleles or anything like that. Now, there are other reasons why we might have a lot of dominant alleles. Like they might be good alleles. And then it would be because selection was favoring them.
So that's assuming no selection, though.
Yeah. So assuming no selection, and all you have is sex, then you can go on and have sex for any number of generations, and it doesn't change the allele frequencies. No. You can just think about that for a minute. Okay, so that's the null model. Now let's start adding some of these evolutionary processes. So now let's add selection to the equation. Here's our standard mouse example. And these mice that live on lava, we would expect the tan-colored ones to get preyed upon more than dark ones. And so we would expect there would be selection in these two populations. And so you can go out there and get a little bit of blood from each of these mice and take it back to the lab and genotype them, and then figure out what are the frequencies of the diploid genotypes in the population. You can also figure out what are the allele frequencies, just by each homozygote gets 100% of its gametes of that allele, and each heterozygote 50%. And you could do this for a bunch of alleles, but let's just say 2. And then you can see how the two populations either conform to Hardy-Weinberg or don't. And you can also imagine like pooling the populations together, and then from the two different habitats, and then you wouldn't expect them to follow Hardy-Weinberg, and they probably won't. Anyway, just imagine mice if you need to have something tangible to think about while we're going through the math. So to build selection into this system of equations that we have, then all we're going to do in this simple case is just say that the fitness of A2 is 1 minus S. And this would be the case where A2 is recessive and A1 is totally dominant, exactly dominant. So if A1 is dominant, that means that A1 looks and has exactly the same fitness as A1, A2. Heterozygotes? So let this be the case. If you let this be the case, and then you kind of crank through the math, then this is what you find. You can model how evolution changes from generation to generation, and you can vary the selection coefficient, S. So if the selection coefficient is 0.1, in other words, 10% of, there's a 10% difference in the in the proportion of offspring that are produced by A2 compared to the other ones. If the other ones made one daughter per mother, then these guys would make 0.9 daughters per mother. So that's what the 0.1 means. Then a new mutation that arose, it would spread in the population, and after about 150 generations, it would be at 90-something%. So that's a new dominant mutation. Yeah. Go ahead, Ruben.
Can you go to 100%, or is that?
It never actually goes to 100% in an infinite population because it's infinite. But once it gets out to 9,999, then the last allele, let's just say it is lost by chance in a finite population. So here's kind of an interesting quantitative result, and that is that it doesn't take 1 million generations to move from very low frequency to very high frequency. It takes a couple hundred generations if you have moderately strong selection. Of course, if you have super strong selection like 0.7, then after 50 generations, it's really up there. Now, this is the case where we have a certain kind of dominance, and the new mutation that arises is a dominant allele. We can also consider the case where the new mutation that arises is a recessive allele, and so on. So here's a couple other cases. The red is the one I think we just did. And then the yellow is a new recessive allele that arises. So when a new recessive allele arises, the first time it arises, of course, it's in a heterozygote, right? Everybody with me? Like if you have a mutation that changes from A1 to A2, and the A2 is recessive, then it's going to be in a heterozygote. So selection isn't really going to see it in the heterozygote. It's just going to sort of be there, right? It's not until you get a couple of heterozygotes that happen to mate with one another, then 1/4 of their offspring would be homozygotes for this recessive allele, and then selection would see it.
Why can't the mutation happen on both at the same time? Or is it just too rare?
Well, it could, but it's very rare, right? So you assume that mutations are happening like 1 in 10,000 cases or something. So the chances of having 2 mutations of exactly the same sort happening in the gamete formation of, both, where you're getting both of your gene copies, it's pretty small. It's negligible, let's say. So this recessive allele, it goes on for hundreds of generations before you just happen to hit enough cases where you have homozygotes. But then those homozygotes, they're selected for, right? They're better than everybody else. And so once they climb up in the population to 20%, then they just zoom to 80. You know, there's no, or they zoom actually close to 100, right? To 99%. It's just that it takes a long time to kind of get off the ground because they're hidden in heterozygous individuals. Actually, the same thing also occurs at the end when we have the spread of a dominant mutation. So if we look at the red line there, the red line, it gets up to 90% pretty fast, but then it takes a very long time after that. And the reason why is because all of those old recessive alleles are all hidden now, hidden away in individuals that are not showing them. They're carriers. Yeah. So from beginning to end, the way that selection there'd be the most response to selection would be the codominant case. If it was equally codominant, where the heterozygotes are exactly intermediate in fitness between the two homozygotes, then we would say that there's maximum additivity in gene expression. Maximum additivity in gene expression. And then it gets a good start right away, and it also goes to completion at the end rather rapidly. I'm not sure everybody understands these graphs. I wonder if I can get over here. Where's my thingy?
The y-axis is the frequency of the allele, right? On the bottom.
Yeah. Yeah. Let's look at the top graphs. So the top graphs are just the fitnesses. Like the 3 bars on the top graphs are the 3 genotypes. A1, A1, A1A2, and A2A2. And so the red one, that's the case where you have a new dominant allele, A1, and it is favored. See, A1 has a higher fitness. A1 has a higher fitness than A2, right? And the heterozygote is just like A1 in its fitness. And the yellow one is you have a new dominant allele, a new recessive allele, and it has higher fitness, right? So A1 has higher fitness than A2, and the heterozygote is just like A2. It's just like the old type, the old-fashioned one, right? And then the one in the middle, the blue one, the heterozygote is halfway in between in its fitness. So if A1, each individual was able to produce 3 offspring, and A2, each individual was able to produce 1 offspring, then A1, each individual would be able to produce 2 offspring, exactly in between, on average, of course. And then you just take those selection coefficients and you plug them into this equation that's buried in some box someplace in the text, and then you just crank through that equation, generation after generation after generation. And it would then model this curve. Mike?
So will recessive ever become dominant? Or?
Not by this process. Now, it could become by other means. You could have a recessive allele become a dominant allele. But that would probably be because at other loci, there's evolution that is making things dominant. Yeah, but by this process, this simplified process, you just have mutations that occur, and they're either beneficial or they're detrimental, and they're either dominant or not, or something in between.
So each boost on that
Curve?
graph, it's a new mutation that came in.
Yeah. In all cases, we're imagining that there's a new mutation at time 0 that comes in, and it's either recessive or dominant or codominant. Yeah. And of course, you could have something that's partially codominant as well. You can do anything you want, right? But and partially codominant, you know, it would be a little bit more like either the recessive or the dominant, depending on, you know, how it worked out. Yeah, go ahead.
Is there an explanation why codominant reaches the fixation more rapidly than the dominant?
Yeah, so the dominant, it doesn't go towards fixation at the top very fast, and the reason why is because the deleterious old allele, it's hidden in heterozygotes. And so selection doesn't have the opportunity to eliminate those gene copies of A2.
So there's more codominant really quickly. So why isn't there more dominant and it's more fit than the codominant?
That's just the way we said it was. So the top thing is like is just what you just imagine particular cases, right? And the top graphs, and then if you imagine those top graphs, then you get the bottom graphs. But I don't know. I'm not saying anything about whether in nature most alleles, new alleles that arise, are dominant or recessive or codominant or whatever. You know, that's another topic. So it could be that most new alleles in nature that are beneficial are codominant. I'm neutral on that. Maybe we'll get to it someday. All right, well, go home and ponder it. Unless you want to talk about it some more right now. Okay, here's the case of overdominance. Overdominance is where the heterozygote is more fit than either of the homozygotes. So if you look at the green bar, here's the heterozygote, and see it has a higher fitness than either of the homozygotes. And if you crank that through the equations, then what happens is you end up with a balanced polymorphism. So as time goes on, you hit some gene frequency where both alleles are present in the population and it doesn't change, which is one of the possible causes for a balanced polymorphism. It's not the only possible cause for balanced polymorphism, but it's one of them. This is overdominance. And the classic example of overdominance is sickle cell anemia in places that have malaria. So in places that have malaria, having the sickle cell allele, it confers greater fitness in heterozygotes, even though in homozygotes there's lower fitness. So if we go backwards, so this might be individuals that don't have either sickle cell allele, and this would be ones that are heterozygotes, and then this one is ones that are homozygous for the sickle cell allele. And if that's the case, then in tropical places where there's a lot of malaria, then these guys are not affected by the malaria very much. The malarial parasite can't get at those cells easily. These guys are anemic, so they're not doing that well, and you might think that it might affect their ability to have children or whatever. Like they're kind of sickly. And that's what people worry about, these days. So if you have sickle cell anemia now in the U.S. where there's no malaria or anything, then you're worried about being this. But if you're in the tropics, then you might be worried about being that. But then there's a benefit to being this, being heterozygous. And so in the tropics, the sickle cell allele is it's in a balanced polymorphism with the regular allele. Yeah, go ahead, Ruben. Overdominance. Oh, it's called overdominance. Yeah, overdominance is where the heterozygote is more or better or something like that than either homozygote. No, codominance is where it's halfway in between. Yeah. Yeah, it only works in the sickle cell case in places that have malaria, because the heterozygote is more fit by virtue of being tolerant of malaria.
There's like a lot more wet, more humid.
Yeah, hot, wet, humid. Any place that it freezes, malaria is not very common. And so in that case, it wouldn't be overdominance, right? If in that case, I think it would be regular dominance, and as a result, sickle cell allele is very uncommon in people that come from the far north. Of course, now everybody breeds with everyone, so sickle cells, you know. Okay, so that was overdominance. And then what do you think underdominance would be? If overdominance is where the heterozygote is more fit than either homozygote, then what would underdominance be? What's less fit? Which one?
The graph slower than the other two. Over time, that allele won't show up because it won't be able to, whatever individual has that allele won't be able to reproduce because it's going to die.
Yeah, so it would be that the heterozygote is going to die or not do nearly as well, right? And so whichever allele you started out being high frequency for, that's probably the one that's going to go towards fixation of. So here's the case where you have underdominance. The heterozygote is less fit than either homozygote. And, as you say, over time, you're going to go to fixation of one allele or the other, and which one you go to depends on the starting conditions. It's going to go towards the one that was the higher frequency to begin with. Not based on 50%, but based on whatever this relative frequency fitnesses are.
What does the overdominance look like?
Yeah, so overdominance, it converges on the intermediate thing. This one, it diverges. So here's overdominance. It converges on an intermediate balanced polymorphism. So if you didn't know what the fitnesses were, and you just went out and sampled, and you found that there was this polymorphism, and it was the same polymorphism everywhere, and you know, it kept on going for generations after generation, then you'd think it was a balanced polymorphism. And one of the possible, but not the only possible explanation for a balanced polymorphism is overdominance. If, on the other hand, you go to a bunch of isolated populations, and each isolated population has is almost fixed for a different allele, then you might think, oh, it's possible that it might be underdominance that's responsible for that. Right? And here's an example of underdominance with these New Zealand mice. So you get normal mice, and then you have these ones that are autoimmune compromised, and they're the heterozygotes. And then the other homozygote, it's not as healthy as a normal mouse, but it's not, you know, as bad off as the heterozygotes. Okay, now neither overdominance nor underdominance is what we would call frequency-dependent selection. It's just what the fitness would be of each of the different genotypes. And so overdominance is where the heterozygote is more fit than either homozygote, and underdominance is where the heterozygote is less fit than either of the homozygotes, and that has evolutionary consequences. But it's not like the strength of selection depends upon the frequency of the allele in the population. So we do have that case, though, and that's called frequency-dependent selection. And here's a case of positive frequency-dependent selection. So positive frequency-dependent selection is the most popular allele is the one that has the highest fitness. And as an allele becomes less and less common or frequent, it has lower and lower fitness. And it's regardless of which allele it is. So here we have in the red the fitness of the P2 phenotype, and in the blue you have the fitness of the P1 phenotype. And you can see as P1 gets more and more common, then that's in the gray. As P1 gets more and more common, it gets higher and higher fitness. In this case, this kind of positive frequency-dependent selection, then if you crank it through the model, you find that a population will go towards fixation of one allele or the other. And a kind of neat example of that is the left and right coiling on these snails. For biomechanical reasons, it turns out that a snail can't mate with another snail that is coiled in the opposite direction. You know, they don't fit together quite right. And so there's a great benefit to being coiled in the same helix as is most common in the population. So if most of the population is coiled with a right-handed coil, then the right-handed snail is going to do much better than a left-handed snail in this circumstance. And then, of course, what happens is the population rapidly goes to all being, right-handed. Similarly, if you had a population that started out where most of them were left-handed, then being a left-handed snail would get higher fitness than a right-handed snail, and then that population would quickly go to being almost all left-handed. It's a good thing this is not true with people. Although maybe it's a little bit true with people. Maybe less so these days. Like what if, okay, you don't listen to this. But I just had an idea. Okay, what if being left-handed conferred like a lot of success in being able to do well in evolutionary biology classes? Whereas being right-handed conferred a lot of success in all sorts of other things in life. And then, then in a little isolated population where everybody studied evolutionary biology, then after a few generations, essentially everybody would be left-handed. And then in some little isolated island where everybody did art, then maybe everything would go to right-handed. Okay, I don't think that's actually true, but whatever. Huh? I'm left-handed, yeah. Are you right-handed? Oh, okay. Yeah, I mean, it's not a single locus or anything, but it is heritable, a little bit heritable. Yeah. And left-handed people, you know, face special challenges, but then after we've survived those special challenges, and we come out feeling superior. So. Okay, so maybe this is a little more interesting. This is negative frequency-dependent selection, and like a great case of negative frequency-dependent selection is the explanation for why we tend to have a close to 1-to-1 sex ratio. Like if you start out a population, say you start a population that's mostly female, then what do you want to be? Male, right? If you start in a population that's mostly male, what do you want to be? Female, right? So assuming that you can only have two sexes and a few other little simplifying assumptions like that, then that would be a great case of negative frequency-dependent selection where the population would hone in on 50-50 because the rarer sex is the one that has higher fitness. Another way, if you wanted to sort of put it in a little bit more kind of accurate terms, It's like in a population that's mostly female, then the best thing to do is to produce sons. And in a population that's mostly male, the best thing to do is to produce mostly daughters. Okay, so anyway, that's not the example the book uses. The book uses these fish that are left-mouthed and right-mouthed. And so these fish, they go and they eat scales off of other fish, which is sort of like parasitizing them, right? They just take a little bite out of other fish. And so ones that have a left mouth are really good at taking bites out of the left side of fish, and ones that have right mouths are good at taking a bite out of the right side of fish. Now, of course, the prey react to this. So if there's lots of these parasites that are right-mouthed, then the prey get really good at avoiding right-mouthed ones, which gives an advantage to left-mouthed fish. And so this is a case of negative frequency-dependent selection where the parasites that are of the rarer mouth side have an advantage. And so what happens is that they hone in on 50-50. Population is always around 50-50. Now, this is another example of balanced polymorphism, right? Because it's a polymorphism, left and right. It's balanced in the population. It goes on and on and on for many generations. And so one possible explanation for balanced polymorphism is that there's negative frequency-dependent selection. Another possible reason for balanced polymorphism would be overdominance. And then there might be some other reasons, too, for balanced polymorphisms. How are we doing? Yep. Okay, so we've kind of marched through adding selection. Now we can add mutation to our equations. I guess these are supposed to be gametes, or maybe they're loci. So you can have a mutation from A1 to A2, and the rate at which that happens would be mu. So and it's another one of these rate things. So the frequency of going from A1 to A2 is mu, and then the frequency of going from A2 to A1 we say is nu. Nu. Nu, with an N. This is with an M, and that's with an N sound. Mu and nu. The Greek letter that looks like a V is nu. And mu and nu could be, they could be about the same. Like, I think the rate of mutation from T to C is about the same as the rate of mutation from C to T in DNA. Or they could be very different. Like, maybe we're considering the rate of mutation from a gene that works to one that doesn't work, and that might have a very high mutation rate. And then the mutation rate from going from one that doesn't work back to one that works might be a very low mutation rate. Because there's lots of ways to screw up a functional gene, but there's not very many ways to put it back together. So in a system like this where you have mutation that's happening, but there's no selection, then what happens is the population evolves over a very long period of time to have some of the genetic variation hanging out in the population. So it hits an equilibrium at an intermediate rate, and that's exactly the same as the ratio of mu to nu, right? But it takes a long time, and the reason why it takes a long time is because mutations are not super common events. Yeah. So this is a neutral case where there's no selection on, for one allele or the other allele. And it's kind of a no-brainer. We'll talk about this more when we start talking about neutral theory. So this is for one locus, but imagine it's for the whole genome. And then you can kind of get some interesting things, because that might be responsible for what's all the variation that we find in the genome that is never expressed into proteins.
Is that like the discrete stuff?
Well, it all is discrete because it's all A, T's, G's, and C's, which are, just, yeah. So it is discrete, but it's a very fine level, right? You have to sequence it to look at it, usually. Well, maybe that's not true. You could look at the proteins. Yeah, you could look at the proteins. People do that. Okay, so let's now, though, make it interesting by adding both selection and mutation. And what happens is you get a selection-mutation balance. So let's imagine that the mutations we're talking about are loss-of-function mutations. So you probably all know that we have thousands of genes in our body, and those genes make proteins, and the proteins do various things. And of course, all that's good and well. But from time to time, each one of these thousands of genes mutates to lose its function. And so then it makes some protein that doesn't work correctly. And if that's the case, then it's going to be selected against. So you're going to have both mutation happening and selection against the new mutants. So imagine we have situations where mutation goes to bad, and that's mu, and then back mutation is negligible. So whenever there's a mutation like this, it's loss of function mutation. There's very little probability that it's going to mutate back to something great, which makes it simple. Then it turns out that we reach equilibrium, and for recessive mutations, the equilibrium at any one locus of this deleterious mutation is the square root of mu over s. And for dominant mutations, it's not the square root, it's just mu over s. S is the selection coefficient. So how strong is selection? Yeah. Now, this becomes quite interesting, I think. This is one of my favorite little topics. One thing is it doesn't really matter that much if the genetic disease is terrible and debilitating and just kills you, or if it's just a little bit deleterious. Because the ones that are terrible and kill you, they eliminate themselves from the population in a few bodies. Whereas the ones that are just a little bit deleterious, like just being short and ugly, they are spread out over many generations. They're spread out over many, many generations, where each generation that the bearers of that allele are, just a little bit less fit. But it's still, it's going to be eliminated from the top population. It's just whether it's going to happen all at once by killing one individual, or it's going to happen over many generations by making each, of a series of fathers and sons have it really hard on them to find mates, and like they have to scrounge around. And they finally, when they're 40 or something, they get a mate. And then they can only have one child because, the mate's old and ugly too. That's... Now, you might think this is no big deal, right? Like, mutation rate, it only happens like 1 in 10,000 individuals, and, so on and so forth. I don't really have to worry about it. And that's sort of true if you're just looking at one locus. It's not that likely that your baby is going to be a mutant at one particular locus. Like, say, you're really obsessed with the locus that causes hemophilia. It's not that likely, that it's going to happen. But here's the problem. There's thousands of loci, right? And these thousands of loci are all potentially causing mutations at a rate of, 1 in 10,000. So it actually turns out that most that most new individuals have a new mutation. Like, we're all mutants. We're all carrying around genetic load. Nobody in this room is perfect. Like, all of you have some deleterious mutations, and you're just, waiting for selection to get rid of them, which might take many generations. But so it's essentially everybody in the population is imperfect at one locus or another. And some of these imperfections, of course, are very, debilitating and horrible, and some of them are just, not having nose that's perfectly straight. I don't know exactly where you call it a genetic disease, but there's lots and lots and lots of genetic diseases that are all very rare and don't have that much to do with one another. There's other genetic diseases that are common, and they're usually common because it's a little more complicated than this, right? The one that they give in the book is a genetic disease that doesn't manifest until people are about 40. Well, by the time they're about 40, they've already had most of the kids they're going to have, right? So it really isn't selected against that much because it's only manifest in old people. Oh, yeah. Yeah, I know. And then the other possibility is that these genetic diseases that happen in old people are actually good in young people. Or they might be good in siblings. Or they might be good in heterozygotes. There's all sorts of other genetic diseases that are not just this mutation selection balance thing. And most of the ones that are super common would be more complicated than just mutation selection balance. So here's the one that they give in the book. It happens when people are about 40, and I guess it kills them off eventually, but they've already had kids. Okay, that's it. Why don't you try to do your peer evaluations, get those going, and then next time I'll finish off this chapter, and I'll also start chapter 8 if you want to try to get ahead of me.